Analysis outside population cells is one of the commonly used methods to isolate and identify cancer stem cells from tumor tissues and tumor cell lines. This assay is convenient, fast, and cost-effective.
This method can contribute to a better understanding of the effect of genes or other extracellular and intracellular signals on stemness properties of tumor cells. Many factors can influence the percentage of SP cells in this analysis, so it is critical to explore the proper conditions before the formal experiment.
Seed tumor cells in a 6-well plate and incubate them in a 37°C incubator supplied with 5% carbon dioxide. When the cell density reaches about 90%, aspirate the culture medium and wash the cells twice with 3 milliliters of PBS. Then, add 500 microliters of 0.25% trypsin EDTA to each well and incubate the plate at 37°C for 1 to 3 minutes. Gently tap the plate to detach the cells, add 3 milliliters of PBS supplemented with 2% FBS to each well, and pipette the cells up and down 3 to 5 times to disperse the cell clumps. Examine the cell suspension under the microscope. If cell clumps are present, pass the suspension through a 70 micrometer strainer. Transfer the cells to a 15 milliliter centrifuge tube and centrifuge them at 200 times G for 5 minutes. Remove the supernatant and resuspend the cells in 3 milliliters of PBS supplemented with 2% FBS, pipetting the cells up and down 3 to 5 times.